right, we're going to hear from Lily from Jefferson. Lily, you're on.
Okay, do you see my slides?
Yes, we do.
Okay, thank you, Sherry and Tamara, for organizing this research session of the conference. Today, I am very delighted to talk about my research on nephrolithiasis in PXE. I am a research faculty and I work very closely with Dr. Vito at Jefferson Dermatology in Philadelphia. Nephrolithiasis is also called kidney stones. These stones are initially formed in the kidney, and when they pass down the drainage system, that result in severe pain, bleeding, infection, frequently requires surgery and transplantation. The majority of the kidney stones are made of calcium oxalate. Okay, I will tell you later why we care about the composition of the kidney stones. And the kidney stones are the most common urologic emergency. So earlier this year, Dr. Vito saw one family with PXE in his Jefferson Dermatology clinic in Philadelphia. So you can see the pedigree here. There are two females that are diagnosed by PXE based on the physical examination of the yellowish papules on the lateral of their neck in both patients. And by histological evaluation, you can appreciate those, especially from this Fancasa stain here, which is a very specific staining for calcium phosphate deposits. And there are those black stained materials in the dermis of the skin, which is a very characteristic feature of PXE. And in the middle panel here, this is a specific stain for staining elastic fibers. So fragmented elastic fibers and calcium phosphate deposits in the mid-dermis makes the diagnosis of PXE. The diagnosis of PXE in both patients are also confirmed by ABCC6 mutation analysis. So in our lab, we have a streamlined ABCC6 mutation detection strategy that we can identify what types and the location of mutations in the ABCC6 gene. So here, we found that in both patients here, both patients carry one mutation in exon 21 and intron 21 border, and one mutation in the exon 27. And the mother and the father, which are clinically unaffected, are carriers for just one of the mutations. So that is consistent with the autosomal recessive inheritance mode for PXE. And we used in silico prediction programs to predict whether those two variants that we saw in ABCC6 are pathogenic. So based on our analysis, we think that both variants in ABCC6 are damaging variants that cause PXE in this particular family. This younger female, the younger sister with PXE, at 8 years old, had 3 years of recurrent kidney stone. And the mother was very concerned about her children and whether the older sibling would develop a kidney stone too in very early age. So we asked the simple question, do PXE patients have a higher risk of developing kidney stones than the general population? So to answer this question, we would like to look at the prevalence of kidney stones in the general population. And then we can compare that in the PXE patients. So to do that kind of analysis, we need two population cohorts. And we used a National Health and Nutrition Examination Survey, short for NHANES survey, to compile data and get the prevalence of kidney stones in the general population in the United States. This survey was conducted by the National Center of Health Statistics of the Centers for Disease Control and Prevention. It is a program of studies designed to assess the health and nutritional status in the United States since 1960s. So starting from 2007, there was one question related to kidney stones that were added to the survey. And the specific question was, have you ever had kidney stones? And all survey participants were 20 years of age and older. So starting from 2007, the survey results were released on a yearly basis. So there are a total of 5 different and independent data sets that have been released up to 2016. And in each data set, there are over 5,000 individuals who responded to this question. Have you ever had kidney stones? So let's look at the data from the 5 independent survey cohorts next. So for the PXE patients, we also asked the same question. Have you ever had kidney stones? That was asked in the national survey. And in addition to this question, we also asked, how old were you when you first experienced a kidney stone? Because in the light of the female patient who had 3 years of recurrent kidney stone since age 8, we would like to know whether patients who had early onset of kidney stone than the general population. So from the PXE International Registry, and we had a total of 563 participants that answered our survey. And among the 563, we had 552 who are aged 20 years or older, because this is because we really wanted to match the criteria and the same question that were answered in the national survey so that our comparisons can be made directly. So this is the cohort demographics of the national survey and the PXE International survey here. So if you look at the red boxed area, so if we split up the participants by age, to 30 to 40, and up to 71 to 80 years of age, and in the national survey, the ages were almost equally represented here. In our PXE national survey, most of our participants are in the age 40 to 60 years of age. And if we look at the sex, the national survey had roughly 50 and 50% male-female ratio, while in our PXE national survey, there are more females. The females represent 78% in our national survey. Now let's look at the prevalence of kidney stones in the general population. So there are 5 independent data sets. And if you look at here, we combine male, female, and we can also look at female-male prevalence separately. The prevalence of kidney stones in the general population is about 9 to 11% over the years. And the males generally have a higher risk of higher prevalence of developing kidney stones than females. So now we can separate the kidney stones by sex, and we can compare this national survey with our PXE International survey here. And if you look at the graphs, so if we look at this all, so the prevalence of kidney stone in the general population is 9.2%. And the prevalence goes up to 23.4% in the PXE cohort, which is statistically significant. If we separate them by sex, female, male, and we see the same statistical difference. So if we separate the groups by age, for every 10 years increase, and you really see a trend of increased prevalence of kidney stone in the national NHANES survey, and which is very similar to what we saw here in the PXE International, except in the age of 71 to 80 years. So our younger patient, who is only 8 years old, had 3 years of recurrent kidney stone. We would like to know how earlier they first experienced a kidney stone. So we compiled our survey results to the question, how old were you when you first experienced a kidney stone? and we generated this data. And our results showed that 18% of PXE patients reported their first episode of kidney stones before age 18. So this number seems to be a big number. However, the national survey does not ask this question. So actually, we really do not know whether PXE patients had early onset of kidney stones. But this is a very interesting observation. So in addition to our survey, there are 2 published papers that reported the prevalence of kidney stones. So for example, here there are 2 papers reported in a French cohort of PXE patients with very different prevalence of kidney stones. This first paper reported 11%, and the second one reported 40%. While we do not really explain the differences, but we think the difference might be due to the geographic area, the dietary preferences, and the constellation of the different age groups of the participants in the survey groups. So as I said in the first slide, the kidney stones are made of calcium oxalate in the general population. And what about the kidney stone in PXE patients? So there are a couple of reports and saying that the stones have been analyzed and they were all made of calcium oxalate, which is the same observation in the general population. As researchers, we have been using animals, for example, the mouse models and the rat models of PXE to study a specific disease process. So we would like to know whether our animal models, for example, the ABCC6 knockout mouse and the rat would develop a kidney stone. And if yes, what is the kidney stone made of? So this is a paper published 2 years ago by a French group, and they analyzed the random plaque calcification, which is considered to be the precursor and the nucleation site of kidney stones. So as you can see the picture here, this is the cross-section of the kidney and in the renal papilla here. And this staining demonstrates the deposit in the renal papilla. And by histological stainings, the kidney stone phenotype really progress with age. So there's very little kidney calcification in the ABCC6 knockout mice at 3 months of age. And you see a very robust phenotype when the animals reach 24 months of age. So in my laboratory at Jefferson in Philadelphia, we are using our ABCC6 knockout rat as another rodent model to study kidney stone in PXE. So in addition to develop skin, eye, and vascular calcification phenotype, which was seen in patients, those animals also develop calcification in the renal papilla. So we did some elemental analysis in the calcified area in the renal papilla, and we found that the deposits were made of, has calcium, phosphorus, aluminum, oxygen, and carbon. So this aluminum is not part of the tissue because this was actually came from the carrier that we have to mount our tissues to. So that aluminum is a part of the metal carrier that we used for the analysis. So the analysis had the calcium, phosphorus, and the carbon. So we asked whether the deposit is made of calcium phosphate or calcium oxalate. Based on the elements analysis, we don't know. So next we did the radar mapping of the elements. So we would like to map where the elements are on the tissue sections here. So see, for example, here, and you see the calcium, the phosphorus, and the oxygen, and they localize with each other. So that suggests that the deposit is made of calcium, oxygen, and phosphate complex. And these carbon maps do not really overlay with calcium, phosphorus, and phosphate. to confirm that the carbon is not present in the calcified area, we did an elemental analysis of an area, for example, here, which is far away from the calcium deposit, and our spectrum shows that the carbon is present in the tissue. So this is the carbon present in the tissue, not present in the calcified area, suggesting that the mineral is not made of calcium oxalate. So this is our research directions in our lab. We are using animal models as models of human PXE to study the disease pathogenesis. We would like to know the progression of the kidney stone formation in those animals. We would like to know the calcium, the mineral composition in those animals. And because as Viola has presented in her slides that the animals are not humans, there are differences in the pathology, there are differences in the phenotypic presentation. And once we have carefully analyzed our animal models, we would like to develop treatments for kidney stones in a preclinical setting. So you have heard from the previous presentations about the paraphosphate bisphosphonase, TNF inhibitors, which all focus on reduced plasma paraphosphate levels as underlying pathomechanism, but there are also other approaches that are independent of paraphosphate. For example, magnesium and citrate, for example, could be tested in our preclinical animal models. So again, this is a collaborative project with PXE International, and I would especially like to give my special thanks to all of the people living in PXE and their families who particularly participated in our kidney stone survey. And I am very grateful to those participants who shared their data with us as scientists. Thank you very much.
Great, thanks very much, Lily. Yes, and I reiterate that, and I've been saying this the whole meeting, that you guys are the critical answers to the very questions you're asking. And we're very pleased that when we got a number of people saying, I have kidney stones, then Lily's lab undertook this analysis and you guys contributed your data, which was very helpful. Okay, questions for Lily? I'm just looking at the chat too to see. Looks like no questions. Good explaining, Lily. Thank you so much.
Thank you.
Okay, next up we have Patrick Thibodeau. Oh, sorry, there is a question from Terry. Go ahead, Terry. Are you muted, Terry?
I'm here. Sorry, I couldn't put my question together quickly. I'm wondering if there's with regard to kidney stones, is there like an onset of when people might get those, or do they kind of have them kind of throughout their life? I guess is my question.
Excuse me, can you repeat your question?
So the question really was, is there a certain period in which these kidney stones appear in people with PXE, or is it variable throughout their life? I actually don't know, but based on the data that we compiled, it seems that it is very scattered throughout. You know, some people will say, oh, I had my kidney stone in my 20s, the other said 40s, 50s, 60s, so they are very different. We got very different answers. Right, great. And Barry, we would also need to get everybody to answer, and we only got a subset of people to answer. So we probably skewed our sample pretty well, although we did ask other questions about eyes and skin so that we weren't just getting kidney stones. But we need a broader survey. We need everyone to answer.
Yeah, and I think maybe just by the data saying they got it in their 60s or their 40s, that would be their onset. So yeah, I think that might have answered it. Thank you.
Great, thank you.